Hello everyone and welcome to today's live broadcast, a keynote presentation using phage to select for evolution of reduced virulence in pathogenic bacteria, presented by Dr. Paul Turner, a professor and departmental chair of ecology and evolutionary biology at Yale University and microbiology faculty member at Yale School of Medicine. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located at the lower left of your presentation window and type your questions into the box that appear on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, notice that you will be viewing this presentation in a slide window. To enlarge that window, just click on the screen icon located on the lower right. If you have any trouble seeing or hearing the presentation, just click on the support button at the top right of the presentation window or use the Q&A button to let us know you're having a problem. Good news, this presentation is educational and thus offers continuing education credits. Click on the button in the bottom left-hand corner and follow the process to obtain your credits. I also want to let everyone know that today's webcast will be available for on-demand viewing through December 2017. Without any further ado, let's join me in completely welcoming Dr. Paul Turner. I will now turn the presentation over to him. Welcome, Dr. Turner.
Hello, I'm Paul Turner from the Department of Ecology and Evolutionary Biology at Yale University and the Microbiology graduate program at Yale School of Medicine. Today I'll present a talk on using phage to select for evolution of reduced virulence in pathogenic bacteria. And I'd like to thank the organizers at LabRoots for allowing me to share the results of some of our recent research with you. So my general interests in viruses stem from a love of genetics, genomics, and evolution under environmental change. And really the breadth of my research program is shown in a slide like this. If you think about viruses and where they exist in the ecosystem, they have kind of a problem. They don't have a lot of control over where they will go at any one time. So a virus is pretty much, it needs to interact with a metabolizing cell in order to replicate. So the protein-protein interactions at sort of the lowest level of biological organization, that proximate interaction can be something that can change on viruses without any of their control. They could bombard with cells that are not able to be infected because of a protein mismatch. So this exerts selection pressure on viruses to change, if possible, to enter a cell type that is changing as well. If you consider multicellular organisms like humans, we have tissues that are not exactly the same throughout our body. So a virus that enters some tissue type and then needs to spread to another tissue has a problem of interacting with a different cell type. So as you move up the level of biological organization to the cell itself, this also presents a challenge to viruses and where they can successfully replicate in the environment. Again, in hosts such as humans with innate immune systems and adaptive immune systems that change over time, this presents an environmental challenge to viruses to keep up with those changes, and they might be able to do so through mutational change, but the point is those environments change on them as well. And then if you consider viruses that infect very different types of organisms that are not at all closely related, an example would be viruses that infect a mosquito and have to replicate in the mosquito midgut before they go on to being transmitted to a mammal such as a human through a mosquito bite. So the necessity for viruses to replicate in two very different types of hosts is another thing that drives the interest of me and the people in my research group. And last, if you consider large-scale changes that are affecting all organisms in the environment, so-called ecosystem-level changes, this is also important for even the smallest denizens of the planet, the viruses. So even though they are very small in size, they can still experience the challenges of a changing environment at the ecosystem level. So this also becomes of interest to me to consider how would viruses keep up if the environment is changing radically at the level of ecosystems. A tool that we often use in my research is called experimental evolution. And this is really a way of studying evolution in action. This cartoon shows you the basic setup of a typical experiment that we might use in the laboratory, where we begin with an ancestor genotype. It might be an ancestral population, but the point is this single entity is used to found multiple lineages across different treatment groups. So in this way, you could look at lineages in, say, treatment A, and the ability for repeated evolution to occur and lineages that are independently evolving in the same environmental challenge. By the same token, you can look across treatments to see whether the actual treatment itself is affecting how these viruses adapt to some environmental challenge. Usually the duration of these experiments can be long or short, depending on what the researcher wants to do. In this particular example, it's shown 100+ generations of evolution. In evolution in action experiments, or experimental evolution, one can keep track of certain traits and how they change over time. So one possibility is to look at this hypothetically, where you have a distribution of some trait of interest that's shown here in green on this hypothetical diagram. And as that population is interacting with an ecological filter or an environmental challenge, you can literally see a change in that trait or the mean of that distribution of trait values through time. And in these types of experiments, we keep track of time by counting generations. We have access, especially in virus experiments, to the entire genome and to examine it through genomics to see how does the ancestral genotype change into those that are adapting within this experiment to the environmental challenge. And you can literally sequence the entire genome pretty efficiently and affordably to look exhaustively throughout the genome for where those mutations are occurring spontaneously that let the populations better adapt to the environment. One final aspect of these types of experiments where we hold a lot of power when you use viruses to conduct them is that we have an immediate inference about the phenotype-genotype associations. That is, we're measuring the phenotypes rigorously, we're looking exhaustively across the entire genome, so we can immediately make some sort of a match prediction between the mutations we see, the phenotypes that change, and try to make sense of what's the real functional role of those mutations. Now, those experiments are not trivial. One might want to then take the mutation or mutations that you observe in your experiment and use further techniques such as reverse genetics to build those mutations in one by one into the wild-type ancestor and prove explicitly that the mutations that were seen in the experiment are responsible for certain functional changes in the mutation. With all that power, my lab group is able to address a variety of research topics. And these range from what could be defined as basic questions to applied questions. So we have a very general interest in how evolutionary processes occur. One could cast that as tempo and mode of evolution and adaptation. And there are lots of handy analogies, especially adaptive landscapes as depicted in this cartoon, to consider as evolution is happening, as new mutations are brought together into genomes to account for fitness changes, one could envision this as climbing a fitness peak to ever and ever higher levels of fitness in the environment. And we can keep track of that in our experiments. More in terms of the ecology and evolution of infectious disease in particular, we cast our experiments in this way to perhaps examine what is the role of direct transmission of some parasite or pathogen between host individuals, such as occurs in a human sneeze, versus what is the role of vector transmission, where, as I alluded to earlier, some viruses and other pathogens have to replicate in very different types of organisms and be transmitted around the environment. So one can cast our experiments in a more deliberate way to gain insight in how infectious disease systems interact with other aspects of the environment, that's their ecology, and also how they change over time, that's their evolution. And last, for a very applied goal, we could look at viruses and both their opportunities and abilities to adapt, as well as the constraints that prevent them from doing so, to gain insight into how do we understand viruses in some very widespread applications such as vaccines to control the spread of disease, but we could even gain insight into viruses and their ability to serve as new applied routes for therapy. So the latter third of my talk deals a lot with that topic. So here's a rough outline for what I'd like to cover. I'll start in a very general way in talking about the evolution of genetic trade-offs. This is something that can be observed in nature, and it drives our curiosity and our experiments, and ultimately I'll show you a very applied significance of considering trade-offs in terms of developing virus applications. A very specific and widespread type of trade-off is called the survival versus reproduction trade-off. So I'll briefly cover how that trade-off seems to operate in all biological systems, including in viruses. And I'll end by considering how evolution thinking, especially those topics I cover upstream, can help us improve virus applications for some important purpose. And the important purpose that I'll focus on is the combating of bacterial pathogens, especially those that are increasing in their antibiotic resistance. So we are running out of options to control certain bacterial diseases. And I'll present how evolution thinking can help us improve phage therapy as a way of using viruses to control and kill bacterial pathogens. So let me start with the evolution of genetic trade-offs. A lot of the research that we do tries to tackle so-called big questions in biology. And there are fewer questions any bigger than this one. Why are there so many species on Earth? A lot of the reasoning behind the answers to such a question deal with the fact that evolution involves compromises. Often when we go and examine evolving systems, we find that they can't do everything well. So essentially, natural selection, as Darwin elucidated so beautifully in his most famous book on the origin of species, he considered in that book how natural selection can operate and allow biological systems to become better adapted to their environments and their environmental challenges, but in doing so, they could experience trade-offs. They could improve in one trait at the expense of performance in another trait. So the graphs in the middle illustrate this basic idea. If you consider two of probably the most fundamental things that you could think of happening in replicating systems, and that is the ability to reproduce, as well as the ability to survive some environmental perturbation. So if a biological system evolves to invest in better reproduction, it could be that that environment causes them to suffer worse survival as a consequence. And by the other token, they could improve in one trait in survival and yet have a liability in their ability to reproduce. So that whole body of work is often covered in a theoretical way. And one of my colleagues at Yale, Steve Stearns, he's one of the main architects of life history theory. And the idea here is that in general, you can look in biology and find that traits cannot be simultaneously maximized, and that you'll often find this fundamental trade-off between survival versus reproduction. So the question is, would viruses, who even are considered as non-living by certain individuals, by definitions of life, do we also see survival versus reproduction trade-offs in these systems? So let me begin by starting with the model system where we examined this question. This is a bacteriophage, or a virus that is specific to bacteria. It's called phage, and it infects pseudomonads. In particular, we often grow it in the laboratory on Pseudomonas syringae pathivars. So these are different species and subspecies, pathivars of this bacterium that grow on certain plant types in nature. We pretty much restrict our studies to the laboratory, but it's nice in terms of this model system giving us a lot of power in the lab because we know a lot about the underlying genetics. It has only 13 kb sized genome, so relatively small, and it has its genes separated across segments within each virus particle. So it has a segmented genome. So those are some of the details of the genetics of the system. More importantly, I'd like you to key in on the diagram showing the lytic infection cycle for this virus. It's fairly typical of a lytic virus in the sense that these small red hexagons are showing you the attachment of a virus particle to a protein appendage called a pilus that's extending out from that bacterial cell. This allows the virus to gain entry and to replicate within the cell using the molecular machinery of the cell. Over time, those virus particles in the cell will mature, and the cell bursts open, releasing those particles that can then go on and infect other cells in the environment. The picture in the middle shows you these little white visible phage particles, in this particular picture attached to the type 4 pilus of the bacteria that are extending out in that diagram. And on the far right, we have a real image of Pseudomonas syringae bacteria on a leaf surface, where I want you to keep in mind that the importance of these pili that the bacteria have are to move or traverse across the leaf surface and to enter the plant through the stomata, through the openings in the leaves. And this allows the bacteria to gain entry to function as a bacterial plant pathogen. So I'll come back to that property later, but for now I just wanted to introduce this system as the phage is called phi6, and Pseudomonas, especially Pseudomonas syringae, bacteria are the hosts that we study for infection. A fundamental problem that viruses exist, that they face, no matter where they exist, is that they are not stable for very long periods. And there may be changes in the environment where they have to experience selection to improve in their stability. So you can consider stability to be a survival trait in viruses. And this is a very measurable phenomenon as shown in this graph. So these are data from a paper in 2008. And here we show that if you just take lysates, or just pure viruses placed in solution of this type, and you incubate them for a very short period of time, only 5 minutes at an elevated temperature, you can contrast those high temperature environments and the stability or the survival of that virus population on average as they feel this extreme stressful environment. So recall that the typical environment temperature that we use in the laboratory is 25 degrees C. And you'll see that if you push the temperature up to a very high level and incubate viruses for only 5 minutes at a time, eventually you'll reach temperatures of 40 degrees and higher, where you can measure extreme mortality or very low survival of the average particles in that lysate. So one could cast this graph as what is typically called a reaction norm for the virus under a stressful condition, and that stressful condition is high temperature. Well, given the experimental evolution method I mentioned earlier, one might be interested in, can these viruses improve in their ability to be stable in these new environments? So the way that you would do that is shown in the cartoon on the right. You could take a pure lysate of the virus, place it in, say, a heating block that was preheated to the correct temperature and the stressful temperature, place the virus lysate in that environment for a very short period, and then plate it under normal conditions. Let the virus then infect the host at 25 degrees C and form plaques, as I'll describe in a moment. In the process of doing this, several virus generations occur through that lytic infection cycle. You can then harvest those viruses, put them back through this experimental condition. And as shown in the circled temperatures on the left, we've done this at temperatures as high as 50 degrees Celsius to see whether these heat shocks lead to improvement of the ability of the viruses to withstand that stressful temperature. Going immediately to some of our published data, these data come from a paper in 2012, where what you're seeing here along the x-axis is increasing temperature, and on the y-axis is this measurement of mean % survival when the viruses experience this temperature. Well, the real take-home message of this graph is that in an experiment where we allowed 3 lineages, shown in red, to experience these 50 degrees Celsius heat shocks for only 5 minutes, periodically throughout the experiment, they did improve in their average survival at the extreme temperature of 50 degrees Celsius. So you may not be impressed by how much they improved, but that average improvement is statistically significant and greater than their counterparts shown in blue that were evolving alongside them under the exact same conditions, except that they were not experiencing the 50 degrees Celsius heat shock. They were experiencing a mock shock at 25 C. Well, the take home here is that at the more benign and yet warm temperatures, we have very high survival observed for those heat shock evolved lineages. In other words, they've broadened their ability to withstand high temperatures, which could be called environmental robustness. But the main point is that they've improved a lot in their survival under stressful conditions, especially relative to their counterparts that evolved for the same period of time but never saw heat shocks. How they did so, it's very important to consider one mutation that came in to all those heat shock lineages that did not come into the lineages shown in blue. And the real difference is the thermal tolerance of a lytic enzyme that lets these viruses both enter the cell. They bring this enzyme with them in order to get into the cell. But as those particles are released, they also use that enzyme to get out. They coat the nucleocapsid, and that's held with each virus particle as it exits the cell. As you can see, the top cartoon, the thermal-tolerant lytic enzyme, has an amino acid substitution that fills what's called a hydrophobic pocket in that enzyme, making it much more durable or stable under high heat. And this contrasts the existing pocket in the lower cartoon for the wild type, as well as in those control evolved lineages. So now we have a very simple and elegant explanation for how these heat shock evolved lineages are able to withstand that stressful environment. They did so with a single mutational change that allowed them to become more heat shock tolerant or thermal tolerant. Now let's key in on the trade-off that comes about when this occurs. On the left now, these are data from an experiment where one can look at the impact of that one mutation alone for the reproductive capacity of these viruses under the normal condition when they're simply infecting cells at 25 degrees Celsius. The blue bar shows you what is the effect of the wild type, in a sense, when competing against itself for mean fitness in this ordinary environment. And by definition, the mean fitness is 1. Whereas the red bar shows you that this one mutational change was enough to cause a huge selective disadvantage, or an S value of approximately 0.25, where there is a huge liability to having evolved heat shock tolerance for the reduced capacity to reproduce under normal conditions. This is clearly a genetic trade-off that spans the two environments, and the amino acid substitution itself can be abbreviated as V207F, meaning that the valine was substituted by phenylalanine at that position. And this causes a life history trade-off between survival and reproduction. Finally, key in on this diagram showing a real picture of what the plaques look like for these two different types of viruses. So the gray in this image is a confluent bacterial lawn, meaning that on an agar plate, one can grow bacteria to a very, very high density. And then the plaques, or these circles on the lawn, are formed when you take a diluted amount of the virus and you sprinkle them on the lawn and you incubate at 25 degrees C. And overnight, you will get cells that are infected by that virus, producing progeny that infect neighboring cells, and you get a little clearing in the lawn that is a circular, kind of a local epidemic. Well, there's a very big difference phenotypically or visually between the plaques that are formed by the wild-type virus. Those are shown with the blue arrow, and these are clear, meaning that all of the host bacterial cells within that plaque seem to be dying as this epidemic is occurring. Whereas there's a very different kind of a phenotype for these V207F, or thermal-tolerant mutants, where the grayness within the plaque indicates that many of those cells are missing, dying as a result of the infection spread of that virus locally in the plaque. In other words, they are visually less productive compared to the wild type. And that's borne out in the bar diagram directly above the image. So these data relate to an earlier study by DePep and Today in 2006, where they took viruses that could all infect E. coli, and they looked at their mortality rate, or survival rate, versus their reproduction rate. And they found a very tight relationship. In other words, it echoes the survival-reproduction trade-off that I introduced. For some reason, if you look at each of these phage types, they have a characteristic ability to reproduce, a characteristic mortality rate, and they fall along this very tight relationship. So essentially, these researchers do not know what are the ecological conditions in the real world that have selected for these viruses to appear wherever they appear on this line. But you can consider the experiment I just showed you as taking any one virus and moving it up and down the line through the power of experimental evolution. So our data do relate to this more broad phenomenon in viruses, especially other phages. We've also shown how it relates to viruses of eukaryotes. But the main point is that viruses do suffer the classic survival versus reproduction trade-offs. So let's return again to this picture that I showed you earlier of how these viruses infect the bacterial cells by attaching initially to the type 4 pili that allow the bacteria to function as plant pathogens. So clearly, if these bacteria are experiencing these viruses in the wild, which they occasionally do, one could ask, well, what selection pressure is going to be exerted on the host bacteria? And if they see these phages, and they can respond through natural selection to improve their ability to withstand phage attack, does that have any obvious consequences? Does that ability to resist phage attack have anything to do with a trade-off? This is shown in the following diagram that indeed it does. So the resistance of these bacteria to phage phi6, one mode of resistance has a huge impact on their virulence. So if you consider in the picture, again, of these virus particles attached to pili that are extended from these bacterial cells, one way for the bacteria to spontaneously mutate to avoid phage attack is to simply shed the pili. They won't have any longer the primary binding site for the phages, so therefore they will escape infection. Well, this is all well and good, and one could observe that in the laboratory, but one would expect that that would have a huge liability in the natural world. And that's because these bacterial pathogens form, in this case, a disease called halo blight, especially in bean plants. So if you consider the loss of the pilus, that prevents the ability for the bacteria to traverse the leaves and get into the plant. Interestingly, if you just put the bacteria directly in the plant, they will happily function as pathogens. So in other words, the pili themselves and their loss, this could be considered a conditional virulence factor because they cannot traverse the leaves naturally, but they're still infectious in the sense of being able to infect and replicate within the plant. But that clearly is an evolutionary trade-off. One way to summarize that is that the phage selection in this particular system can lower bacterial pathogenicity by experiencing the phages, and if the result is that mutants of these bacteria are favored who shed the pili entirely, well, that is one way to escape phage attack, but it makes them necessarily worse pathogens. All right, so the way that I'm going to link this to our current work in phage therapy is that we had been working in this system for some period of time, noticing this effect of phage selection on lowering the bacterial pathogenicity, and we thought, how might this phenomenon inform our ability to choose better candidates for phage therapy? So we can ask the question, can phage therapy exploit evolutionary trade-offs? So let me begin by reminding the audience of the very familiar problem, an unfortunate one, is that antibiotic resistance is spreading in bacterial populations. We've observed this for decades and it's still occurring. This means that first-line antibiotics are becoming less useful as the prevalence of multi-drug resistant bacteria increase on the planet. These MDR, or multi-drug resistant bacteria, and a good example of them is Pseudomonas aeruginosa. It's particularly worrisome for cystic fibrosis patients, severe burn victims, and in general for any immune-compromised patient. The problem is that Pseudomonas aeruginosa is very readily found even within our own homes. It's in soils, it's in kitchen sink drains, it's everywhere around us. So opportunistically, this pathogen could enter the human body, and depending on your physiology or your disease state, it can be very worrisome for your health and sometimes deadly. The real problem with antibiotic resistance spread in this system is that these bacteria have a complex of proteins called efflux pumps, as shown in the diagram here. A very simple way to conceive of this is it is several proteins, roughly 3 of them, that function as transport protein complexes, that if the antibiotics permeate within the cell, they're effectively pumped out through these efflux pumps. And these function very generally. I mean, not only do these efflux pumps function in a variety of things for the cell itself, including disease-related things like host colonization for certain tissues, avoiding host immunity, and forming tight assemblages of bacteria called biofilms, but they simply can easily function to pump out antibiotics across a wide range of antibiotic classes. So the efflux pumps, a little more on them, they're typically chromosome encoded and genetically conserved. They're generally found in gram-negative bacteria. And as I alluded to a moment ago, they are a major determinant of resistance for different classes, including aminoglycosides, macrolides, and tetracyclines. So within Pseudomonas aeruginosa, this is a very worrisome problem that you see the increase of multi-drug resistant genotypes in the system, and they do have important health relevance. What are we going to do about it? So many people around the world are actively researching to find ways to treat bacterial infections aside from the old traditional routes. If we find that antibiotics are failing, what other options might we have? Well, interestingly, we have an old idea that's been still studied in certain parts of the world, but wasn't ever very popular in Western medicine. And that is a concept known as phage therapy. The Russians were the first ones to develop this idea, but it also was popular in countries such as Poland long ago, and even predating the discovery of antibiotics accidentally by Alexander Fleming. So the idea here is that you would find a phage that is specific to your disease bacterial target, and you essentially develop this phage as a drug. So it will find and kill the bacteria that it is supposed to kill, and be incapable of killing other bacteria, including those that are beneficial to your health and might exist within your microbiome, now that we know much more about that phenomenon. But as long ago as World War II, Russian soldiers were sometimes outfitted with a small vial of phages that they might use in the field if they were wounded. Why? Because their wound could be colonized by a bacterial infection that might hurt or even kill them before they were able to reach someplace where they could get some help from a health physician. So that's one age-old example, but even recently, when you consider certain bacterial infections and diseases, especially cholera, we know already that clean water and rehydration therapy is essential for trying to save a cholera victim. But if you administer phages that are specific to these cholera bacteria as well, this can also hasten their ability to recover from a cholera disease infection. So we have some good news, but frankly, we need more data to figure out how beneficial is phage therapy, and would any old candidate work in terms of finding a phage that will do the job? Well, we didn't think so. We wanted to look specifically for certain phages with certain properties that should be able to do the job better. So consider the following. If you think of a genetic trade-off as being widely spread in nature, and I tried to convince you of that earlier, how might it impact our ability to improve antimicrobial therapy? Here again is one of those typical cartoons for a lytic phage infection. And this infection cycle, if you have phages infecting bacteria, as I mentioned in the 5-6 and Pseudomonas syringae example, what you expect is the following: that if possible, these mutants in the bacterial population will be favored that are incapable of being infected by the phages. Here I'm showing a change in the exterior proteins of the cell on the right, now shown in blue, and this is presenting the incapable ability of these phages to get in and kill the bacteria. Well, suppose what came along with that was somehow an increased sensitivity to antibiotics. In other words, as a result of changing through mutation to avoid phage attack, there was something that made these cells more vulnerable to antibiotics. That would be terrific for phage therapy. And the reason is that it would first give us an option to do the antimicrobial therapy itself. And also, if it is sort of reinvigorating our ability to use antibiotics that are no longer useful, then we can extend the lifetime of current antibiotics in our so-called arsenal. And this is the kind of time that we need in terms of the spread of multi-drug resistant bacteria on the planet. We may find new solutions, but ideally this kind of a genetic trade-off felt by the bacteria would let us go back to an old solution and revive that at least for some period of time. So this is exactly what we found, and I'll go through the next series of slides to prove to you that this was the case. We found in nature a phage abbreviated as OMK01 from the family Myoviridae. And these lytic phage were found to bind to the outermost proteins for certain efflux pumps, especially OPRM, that extends beyond the cell surface and is available as a phage binding target. We confirmed this using a series of knockout bacterial genotypes from a collection obtained from University of Washington. But the point here is that the phage was able to infect all of those bacterial mutants except the ones that had a knockout of the OPRM, meaning that it did not present this binding target on the cell. So we discovered and sequenced this virus relatively recently in 2016 here at Yale. And it has some very interesting properties. When the phage-sensitive bacterial population that is normally able to efflux antibiotics, they are killed by this phage. In other words, the phage binds, enters, and replicates and kills the bacteria. The phage-resistant mutants have an impaired ability to efflux antibiotics, meaning that they are now vulnerable to some drugs that are more or less useless in the system, and they become useful again. Where we found this phage is interesting. We simply looked in water samples in a nearby lake called Dodge Pond here in Connecticut. So we had no idea whether the phages would reside in this lake that had these particular properties. But the point is, because we brought them into the lab and put them through a particular screening process, we were able to find these very intriguing phage candidates. So a little bit more on how this phage operates and what we observe when we bring it into the laboratory. So again, the evolution of Pseudomonas aeruginosa resistance to this phage causes sensitivity to certain drugs, or the so-called desired trade-off that I presented earlier. So what does that actually look like when you measure it in the laboratory? Consider this agar plate, which has a confluent lawn of bacteria on it, and one could place a small disc containing a drug at some high level of density that then spreads out on the plate. And whatever level of that drug that the bacteria can grow to, they can sometimes grow up right to the edge of, in this case, a Kirby-Bauer disc. So these multi-drug resistant bacteria pretty much shrug off the effects of that drug, and they have no zone of inhibited growth. But at the lower portion of this picture, it shows you that one mutational step away, when these bacterial populations are exposed to the phage, those that are able to survive because they are spontaneous mutants, they have a very clear and different minimum inhibitory concentration. In other words, you readily observe this large killing zone, indicating that they are now more sensitive to antibiotics. Looking a little closer at this phenomenon, I'm going to present a table that summarizes a lot of information, but it was presented in our 2016 paper. Let's begin with considering two antibiotics for which there is very rock-solid evidence from the literature and prior experiments that tetracycline and erythromycin, from these two different classes shown in the second column, tetracyclines and macrolides, we know that efflux pumps function to allow Pseudomonas aeruginosa to be resistant to those drugs. So if you look at, on average, the isolates for a variety of bacterial strains and their minimum inhibitory concentration, when you expose them to the phage and you find resistant mutants to the phage, you get a very different MIC for these strains, shown here as an average value. And one can look in the final column at the fold increased drug sensitivity, and you find that this is a very impressive change that is exerted completely by the phage being in the environment and causing either the bacteria to die, or for those that survive to be much more vulnerable to these two drugs. Now, one can consider 4 other drugs from 4 other, in this case actually 3 other classes, and you'll see the changes in MIC and the fold increased drug sensitivity. And you may not be as impressed by those numbers. They're not as large of a change. However, I'm showing with the asterisks that at these levels, the change is relevant from a clinical resistance to susceptibility breakpoint, meaning that we have now, for 6 total drugs that I'm showing you in this table, very good evidence that, in this case, these 4 drugs in the middle, we know that efflux pumps function most of the time in determining resistance, maybe not all of the time, as in the top 2 drugs on the table. But now we've got 6 different instances for a variety of bacterial strains, all Pseudomonas aeruginosa clinical isolates, that this is working very well when they feel the exertion, the selection pressure exerted by the phage. Now last, as a control, consider a drug like ampicillin. So this is in the class of penicillins where multi-drug efflux pumps do not function in terms of determining the resistance. Well, not surprisingly, we got no change between the MIC pre and post-phage exposure. So that control, in a sense, worked very well, and it is very consistent with the rest of the data that I presented. So the summation of this is that only drugs exported by multi-drug efflux pumps are affected in this analysis. Let's look at one more data set that is so far unpublished, but if one wants to bring a little more reality into this, you could consider that Pseudomonas aeruginosa and many other bacterial pathogens, something else that's very difficult in terms of even delivering antibiotics, if they did work, is that they form these structures called biofilms, which are very resilient to anything getting into this tight network of cells and the matrix that they exist within because of what's extruded from these cells binding them together. So shown in this diagram, if you expose a biofilm to antibiotics, shown in one color here in red, you don't expect them to get in. In other words, the biofilm is resilient to that. But some phages, including OMK01, have the ability to get into the biofilm and destroy it and expose those cells, some of them, to antibiotics. So you have a synergistic effect. So these unpublished data are very interesting in the sense that they suggest the interaction between the phage and this antibiotic have a better killing power than either the phage or the antibiotic alone under many circumstances. So what we did was to consider 4 very popular types of material that might go into the human body through surgery, and increasingly so in terms of popular things that are put into the body to help make our organs functioning again when they've worn down. So if you consider the black bars in this diagram, this is what happens when the bacterial densities in these biofilms are exposed to certain antibiotics, ceftazidime and ciprofloxacin, and the 2 other colored bars here next to them, that basically you see no effect of these antibiotics when you expose the bacteria in these biofilms on these different substrates to those 2 drugs. However, in each set of data, the bar on the left, shown in darker gray, this is when you take phage OMK01 and expose those biofilms on these substrates to the phage alone, you get a much reduced density of cells as a result. And that's very promising from the action of the phage alone. But now consider, especially those bars that have asterisks over them, this is when you combine the phage and the antibiotic to get a better and a more synergistic killing. So these are also very promising data because they suggest that if a bacterial biofilm for a multi-drug resistant bacterium is forming on some artificial part of the body that through surgery has been altered, these are great substrates for biofilms to form, and now we're demonstrating that this synergistic effect between the phage and the drug might be enough to clear such very tough and resilient infections, and that would be excellent for the outcome for the patient. So what do we do next in this system? I'll end by talking about some future work and applications. First of all, the trade-off in the table that I showed you and our other work is very promising in the sense that we've observed the same phenomenon for a variety of laboratory model strains, for clinical isolates from a variety of sources, and even for multi-drug resistant bacteria that are taken directly from the environment. They might be in a human home or an estuary and still be multi-drug resistant. And in a sense, we get essentially the same outcome. So the objective now is to look even wider, look at very large clinical isolate repositories to make sure that this good news extends even beyond the large number of strains that we've looked at. With the approval of the U.S. Food and Drug Administration, we are seeking to treat chronically infected human volunteers. We've actually done that with one volunteer patient who had an aortic arch replacement. So this was an artificial part of his heart that his physicians later found out was colonized after surgery by this opportunistic pathogen, Pseudomonas aeruginosa, such that by the time he was feeling very ill effects and they observed this very massive biofilm formation around his heart, there was very little to nothing they could do for him because he happened to be of an age where if they took that part out and put another artificial part in, he probably would not have survived that surgery. He volunteered for the experimental treatment using the phage that I described, and we are currently in the process of publishing his successful case study. Already, if you want to hear more about it, you can find out through a Public Radio International piece that we did for Science Friday in 2016, and various other radio spots and some online news describing this case study. So it was a very promising outcome, and yet, in a sense, we want to take a big step backward, that we were fortunate that we helped a human volunteer, but ordinarily, somebody would get to this point after a large amount of, say, mouse studies in the laboratory in a controlled way. And that's exactly what we are attempting currently. So our objective is to look at the safety and efficacy in a mouse model for lung pneumonia in immune-compromised patients. So this can be mimicked in mouse populations in the laboratory. We were fortunate to receive an NIH Preclinical Services Award in 2017, and these experiments are underway. I don't have very many results to show you so far, but I can give you a little glimpse of something that is related to these experiments and some good news about how this phage operates when it's inside of the largely black box of a live animal and trying to cure these infections through this therapy. So what this graph shows is one can consider this phage that we grow up in the laboratory on a typical model strain, PA01. And in the sense of these mouse experiments, the pathogenic strain that is actually used is abbreviated as UNCD. Well, this graph is showing you that how well does this phage grow in the laboratory on this model strain versus the one that's a pathogenic strain. And the gray points on this diagram show you that the phage grows much better on the model strain when we produce it in the laboratory compared to when we challenge it to grow on that pathogenic strain. Whereas those data shown here in red and blue are from mouse tissues, from infected mice, experimentally infected mice with the pathogen that were then treated with the phage and saved through the process. We find that in a very short time period, these are only 3-day long acute pneumonia models in the laboratory, these phage taken from the live animals now grow orders of magnitude better on that pathogenic strain than they do on the host strain that we grew them up on before we provided them to the researchers who did these experiments. Okay, so the main point here is that this is an amazing amount of phenotypic improvement for this phage in this system. It's a DNA phage. We don't think that it has a particularly high mutation rate, just a standard mutation rate that you would see in a DNA phage. And yet, in this circumstance, in the therapy itself, the phages dramatically improved in their ability to kill the target bacteria. That's good news for phage therapy as well. So our broader objectives that I'll end with is to continue the kind of clinical applications that we seek to conduct in a wide variety of systems: hospital-acquired pneumonia, cystic fibrosis-associated pulmonary infections, catheter-associated UTIs, and burns. So we are now seeking approval to do investigational new drug status for this phage and to help a lot of people and to examine how this phage operates in a real clinical trial, and the mouse experiments help get us closer to that goal. Very last thing I'll say is that honestly we thought we would make faster strides in agricultural settings because there are fewer regulatory hurdles there. So this is a reminder that in a lot of the food that we make on farms, increasingly these are on large and large farms to feed a hungry world. And an example would be shrimp that is farmed largely in Asia and then distributed around the world. Well, there can be outbreaks of bacterial diseases in these farm settings that destroy the shrimp or make them unpalatable to be sold to, for example, stores and restaurants. So, of course, industry in those types of settings or plant agricultural settings, they would be interested in the ability of phages to solve their problems with multi-drug resistant bacteria as well. So we hope to partner with agricultural, industrial settings to try and examine how this might actually help food resources as well. So I'd like to end by acknowledging the individuals who did the work for the studies that I presented. In general, I'd like to thank my lab members. My current lab members are shown here in the picture. And the main architects for the phage therapy work that I described. I'd like to thank Ben Chan, who is a research scientist in my laboratory. So he's a senior postdoc working with me. John Wirtz, he's also here at Yale in the Coli Genetic Stock Center. He's a longtime collaborator of mine and worked on these experiments with us. And I'm especially grateful to Dr. Deepak Narayan, who is at Yale School of Medicine, and he was the surgeon who collaborated with us on this work. Just above him, we have a picture of Ben and me thanking the patient who volunteered for this study and the volunteer therapy, experimental therapy that we conducted. Thankfully, this worked. He is back at work, so we are visiting him in his workplace, and you might be able to make out that we gave him a thank you card as well as a phage plush toy. So I'm not trying to sell those items, but we had a lot of fun thanking him, and he really was very brave to undergo this experimental treatment. And last, I'd like to thank the funders for the work, NIH, NSF, Yale itself. Project High Hopes has been a great funder of our work in the past few years, and the NSF Beacon Center for Evolution in Action. And I'd like to thank you for your attention. I look forward to answering any questions via email that you might have for me, and I genuinely hope that you enjoyed the presentation.